Hello, everyone. So today I thought I'd take you through my methodology that we used in the field and touch on some results that we came up with. They're fairly preliminary. We just finished the counts. Luckily, most of the taxonomy is done. So let's get started. So I thought I'd start us off with a chuckle. Gary Larson actually influenced me into science when I was a young man, and apparently I wasn't the only one because he had an owl louse named after him. So what can invertebrates tell us about the nature of the invasion in Humboldt Bay? Insect and invertebrate communities are essentially reflections of the resources available to them in a given habitat. And in salt marshes, these organisms are typically very specialized with regards to what they consume or prefer as refugia. So taking that into consideration, they should make excellent vehicles for comparing what we consider to be trophically impaired habitats to hopefully fully functional trophically functioning restored areas. So the first question that I want to address is the basic one of what organisms are occupying restored and invaded marshes, and particularly in the Mad River Slough. That was a good study area for us. The literature is really lacking on this, and especially using the method that I utilize. It's never been done before, so it's pretty radical, but not really. So the second follow-up question is there a difference in the community composition between restored and invaded areas? That's a pretty fundamental question to answer, and we expect that there would be based on what I mentioned before. So we have a total of 4 study sites, 2 invaded and 2 restored. The invaded sites are up near the Lanphier Dunes headquarters, and the restored sites are further down by the Mala'el Dunes unit. So our invaded sites are just north and south of Lanphier Road. These are a couple of smaller islands, probably a ½ acre in size or so. You can see here the distribution of sampling points across these islands. Principally selected because of ease of access. These are right off the road, although we did get stuck on an island and had to be rescued at one point. Not paying attention to those tides. So these are completely dominated by Spartina, and these were also selected because the restoration has been so effective along the Mad River Slough that these were the only fully invaded sites left that hadn't been touched by restoration efforts. Our restored areas, these were restored probably 3 years prior, around there approximately. They were allowed, the native vegetation was allowed to recolonize. These are primarily dominated by a mixture of Disticlus and Salicornia in various zones, as we'll see in a second. So our sampling scheme, this is a photo at high tide of our invaded North Island. 25 points were randomly allocated to each site using ArcMap GIS, which is a wonderful tool for this sort of thing. These points were then loaded into a GPS unit, and these points were navigated to in the field with those GPS units. Once we got to a point, we assessed cover class of both vegetation and soil surface. And for those of you not familiar with Dobenmeyer, it essentially just breaks the classes up into 6 percentage categories. And we did that for vegetation and soil. Once that was accomplished, we did our vacuum sampling. This was done 25 times per site in as many habitats as were available on the site for a total of 100 samples that we gathered. After this, we sampled for aerial and vegetation top dwellers with a standard sweep sampling. This was only one sample per habitat per site, which doesn't sound like a lot, but 100 sweeps is a lot. That ended up being a lot of critters, as we saw. So the first habitat that we sampled in is low marsh, and this is typically dominated by Salicornia, typically a dense monoculture, very easy to distinguish from the other zones, just primarily on vegetation color and the time that we sampled, which was in early September. The Spartina marsh, of course, that we've seen today, very dense. These are essentially monocultures, very sparse understory. This was typically like an algae, or if there was any Salicornia, it was just a few unhealthy strands of it, but not much else going on. The mixed marsh or high marsh, definitely dominated by Disticlus spicata. Again, very thick monoculture. these made really nice clear-cut habitats to sample in because we can kind of trace back which organisms are utilizing these. So the first method was actually a converted leaf blower that I used to vacuum these critters up. And I used a jelly strainer as a filter in the end of the tube, which I clamped on, which was really effective because it caught everything down to the smallest of the soil mites. So it wasn't anything much smaller than that we're interested in anyways. So once we get to a point, this is our fancy containment vessel here. The vacuum was run thoroughly over everything in the container for 30 seconds, which doesn't sound like a long time, but we're only in an area about this big, so it seems like forever when you're doing it. The debris and organisms that were collected with this were bagged and labeled for lab processing. So we've got a pretty neat little, oh, video not available, not gonna work. Anyways, shows it how it works. So I'm either gonna get a sponsorship from Ryobi or a cease and desist letter because it destroyed the vacuum. So here's the sweep sampling method and one of my associates out in the field. This was done by 100 sweeps moving forward while avoiding the previously sampled vacuum points. And this is pretty typical of what we'd find, just hundreds and hundreds of flying organisms, typically mostly Dolichopodid flies, but overall mostly Diptera. So once we got into the lab, the material was put into stacked mesh trays. The vegetation was thoroughly rinsed. Each tray was successively searched. The top two, anyways, were successively searched in this big white tub, which made searching for the critters much easier. And once you got down to the bottom tray, the finest material was then put into a petri dish, and these were examined under a dissecting scope, and the organisms were separated from the debris and then stored in ETOH. So this is pretty typical of what we'd see. This is a sample. You'd put the sample, try and evenly distribute it amongst the four quarters of the petri dish, starting in a random section and working off of a taxa list that I'd compiled while we were doing the sorting. You work down the taxa list in the first quarter, and if any of those went over a count of 100, you went ahead and extrapolated that out by multiplying it by 4, because counting every single organism would just take way too long. If it was less than 100, you'd tally the entire sample. Then once you worked all the way through the taxa list, you go back and research the sample, make sure you didn't miss anything. So we were very thorough at this. Next, all the data went into an Excel spreadsheet. These will go towards diversity indexes and also an ordination, which are pending. So here's an example of the over 100. You wouldn't want to sit and count all these. It'd take months. So the community composition that we found at the invaded sites looks something like that. There's 3 organisms that really kind of stand out here. And one, that's the abundance of mollusks, close to 16,000 mollusks in the invaded areas. We had almost 1,300 armadillids, which are an isopod. And then those were followed by the tilitrids. These are an amphipod. Little shrimpy guy, actually. Smells like shrimp when you're working on them. So the restored areas, here's what the distribution looks like. Very different looking distribution. Some important things to remember here. Less than a fifth of the total of mollusks were found in the invaded area, or restored areas, sorry. Quite a few more tilitrids. These guys were much more abundant in restored areas. Hemipterans, lots and lots of hemipterans, and lots of soil mites of various forms. So comparing these side by side, we see a few more things stand out. Number 1, difference in the mollusks. Number 2, big difference in tilitrids. 3, of course, big difference in hemipterans, big difference in armadillids, and also in the soil mites. So the organisms that we found out here, the delphacids, these guys are a hemipteran that were really prolific, especially at the restored sites, and these guys are actually a phloem feeder, and I'm willing to stake money on the fact that they're a specialist on Disticula spicata, because this the delphacid family is definitely more grass phloem feeders. So in the low marsh, we found lots of tilitrids. These guys are essentially a middleman in converting detrital biomass into forms available to higher trophic organisms. So these guys are really big time fish food, bird food, everything else. And like I said, I mean, they really smell like shrimp. You kind of get hungry when you're looking at them under the scope. I don't know, looking at them too long. So our soil mites, the guy on the left is a predatory form of the soil mite, with the two on the right being fungivores / detritivores. Also in the low marsh, Littorina subrotundata. This guy is kind of beat up all over his range, mostly due to habitat loss. Up and down the western seaboard, this guy's just really shrinking in habitat, and its numbers are suffering. So in the Spartina marsh, this is a photo of high tide. And one thing you'll notice is that the tops of the Spartina are still exposed, unless it's one of those 9-5 tides. And what this essentially translates to is this provides an extra habitat niche that's not available at the restored areas. So typically you'll find this guy, the Tetragnatha. This is a long-jawed orb weaver that we just did not find in the restored areas at all. And this guy dominated the tops of Spartina. You can walk out in a Spartina field, and if you hit the light just right, their webs cover everything. A lot of this guy too, Myositella. This guy was very, very numerous at all the sites, comprising 75% of the total catch. And I think we got close to 18,000 snails. So this guy's all over the place. And he's been in Humboldt Bay for, I think, 100 years or so, they suspect. Also in the Spartina marsh, the armadillids. These guys are a detritivore. Something about the Spartina that they like. I couldn't tell you what as of right now, but I'm looking into it. So the interesting take-home message from this is this ratio of Littorina to Myositella in the restored and invaded areas. In the invaded sites, Littorina only comprised 23% of the total, while in the restored areas it comprised 45% of the total. We'll get into why that probably is here in a second. But that's a big take-home message, this. So management considerations: is there competition between Myositella and Littorina? Berman and Carlton in '91 put out a study, I believe it was either in Oregon or Washington, basically said that the invasion of Myositella wasn't at the expense of Littorina. But I have to kind of question that, because these sample areas were so small that we were working in. Just as a matter of physical space, these guys probably are going to be competing. But I'm not totally sure. That's an area that could probably be researched a little bit more, especially in Humboldt Bay. The higher abundance of tilitrids in the restored habitats is probably due to the Salicornia mutualism pointed out by Brebsky and Irwin in 2001. The tilitrid will essentially burrow into the soil, aerate it, deposit urea, while providing habitat and cover for the tilitrid. So it definitely encourages these guys. So in conclusion, the results of the trial seem to indicate that Spartina invasion facilitates high densities of the invasive mollusk Myositella, while removing it and allowing native vegetation to return probably helps the vulnerable native Littorina. The study, I think, demonstrates the much supported notion that Spartina is detrimental to the native biota of Humboldt Bay, and therefore supports the active removal. So I want to thank everybody that's helped out, Andrea, for providing funding, and all my project assistants. We got through an enormous amount of samples this semester. We got 100 samples completely counted and picked through, which was an enormous task. So I'm taking everybody out for beers on Saturday. And of course, thanks to Allison.